All right. Okay.
Yes.
Take it as a yes. All right. Yeah. Thanks a lot, everyone, for coming to the presentation. Let's change the heading a bit. So it's now called Hunting for Hidden Chemical Treasures. But before I go on to the presentation, some of you might be curious about the picture in the background. It's actually a view from the Mendota Terrace here at Madison, Wisconsin, where actually ISMB will be held. So if any of you are coming here in person for ISMB in like 2 days, it's a good view and I hope you guys can catch it. So that's nice. So let's get on to the presentation. So my motivation through this presentation is basically to encourage everyone or motivate to see how amazing marine natural products are, especially marine organisms like sponges and the diversity of natural products that they harbor. So let's get into it. The hero of our story is this sponge, Forsythia. And just to show you what this sponge looks like in its natural environment, I want to show you this short clip, which was actually taken from the place where the sponge was extracted in the Gulf of Mexico near Florida. So it's just amazing to look at this sponge in the whole vast ocean harboring natural products. So why is this sponge interesting? Well, the sponge produces a compound called Lasonolide A, and Lasonolide A is actually shown to have really potent anti-cancer activity in nanomolar concentration, which is pretty cool. Plus, it has a really unique mechanism of action, which is ideal for anti-cancer drugs, as we want the resistance to develop as late as possible. However, everything is not unicorn and rainbows. The supply of this compound is limited. The sponge is the only known natural producer of this compound. And as you can know, or as you can imagine, it's just not sustainable to use all the Forsythia sponge just to extract Lasonolide A. It just doesn't work. So a plausible thing would be to call the organic chemist in the house and ask them to synthesize this. So people have tried this, but it's a really long process and the yields are not amazing. So what do? Well, it's been shown that whenever sponges are producing a marine natural or producing a natural product is generally the symbiotic bacteria that's associated with the sponge that's producing it, and not the sponge itself. Furthermore, if we look at the structure of Lasonolide A, the beta branches and the tetrahydropyran ring suggests that it's being made by a trans-AT polyketide synthase pathway. And such pathways are more commonly found in bacteria. So we hypothesize that probably this compound, Lasonolide A, is being made by the bacteria and not the sponge. And we did metagenomic sequencing to find out the pathway and the bacteria. And our goal was if we can find the bacteria or the pathway that's producing Lasonolide A, we can do heterologous expression or cultivate the bacteria and then in that way increase the supply of Lasonolide A. So we did the sequencing, we got the reads, we assembled the reads into contig and binned the contig into metagenome assembled genomes. And we got a graph that looks something like this. I know it's a pretty complicated graph, so let's break this down together. The two axes here are the dimension reduced axis of a 512 chemofrequency table. And each dot on this plot is a contig, the stuff that you get after assembling the reads. And it's been shown that contigs belong to the same genome would cluster close to each other. So I analyzed this data and I identified the contig making Lasonolide A, and it was here. And I also was able to find out the genome that's this contig is associated with, and it was here. And as you can see, the contig LAS-PGC is not that close to the genome it belongs to. But I mentioned here on the right that contigs belong to the same genome would cluster close to each other. So what's happening? Well, we think that Lasonolide A is being horizontally transferred. So, and the reason I think it's being horizontally transferred and not just incorrectly binned is because we verified the binning using assembly graph, mapping of parent reads, and even doing PCR. So we know it's connected, but the frequency is different, and that's why we think it's horizontally transferred. So this was pretty cool. If we look into the Lasonolide gene cluster or the contig having Lasonolide gene cluster in detail, it looks something like this. each arrow is a gene. And I was actually able to go through, each gene in detail, annotate each domain, and actually identify the entire biosynthetic scheme for the synthesis of Lasonolide A. So it was a pretty exciting moment. We found out that this context is actually making Lasonolide A and not some junk context. Plus, this pathway was actually found out to be a trans-city polyketide synthase pathway, which was pretty cool and matched our initial hypothesis. Right, so one thing that I have not mentioned about LASPGC is that it's not just present once in the genome, it's actually repeated 3 times. And the 3 repeats, the first hint of the third 3 repeats was from the 3x coverage of LASPGC as compared to its host genome. And coverage can be taken as a good proxy for abundance. So said differently, the abundance of the LASPGC was thrice as the genome. And once we found out the coverage is 3 times, we verified it using PCR, we verified the connection at 2N using PCR, assembly graph, and mapping of parent reads. So we actually able to confirm that, yeah, it is actually repeated 3 times. So that was pretty exciting too. And then the next question I wanted to ask, like, yes, there are 3 repeats, but is there any differences? So I was able to find 3 insertions and 2 single nucleotide polymorphism between the 3 repeats. Fortunately, the insertions are in the multiple of 3, as you can see. So they're not causing any frame shift mutation, which was pretty nice. Plus, the insertions were not coding for any PKS domains or any genes that we know. So they're not affecting the translation of Lasonolide A in any known way that we know. SNP1 was coding for the same amino acid, that is tyrosine, after the substitution, and SNP2 was outside the coding region. So as far as we know, these differences are not affecting the translation of Lasonolide A in any known way. So I've been talking about the LASPGC a lot, but what about the bacteria that actually has this context? Well, I showed initially this bacteria is present here in the BHT graph. And when I analyzed this, I actually found out that this bacteria actually belongs to a novel genus in the phylum Verrucomicrobiota. And this bacteria is a completely new bacteria. It's not been discovered before, and it's a new genus. So the moment I realized that, wow, this is a new bacteria that's not been discovered, let's name this thing. Because as scientists, we all want to discover things and name stuff, right? Like Odin from the first presentation. So I decided to do that. I named this Candidatus thermopylae lasonolidus, and I'm going to tell why. So Candidatus, because it's still uncultured, we identified using shotgun sequencing. Thermopylae is actually a tribute to the 300 Spartans who fought the Persians at the Battle of Thermopylae. So there were 300 Spartans who battled Persians, and our pathway is repeated thrice. The Spartans fought to protect their kingdom from the Persians, and we hypothesized that the bacteria is producing Lasonolide A to protect the sponge from any predators. So here's the analogy there. And if any of you still don't remember Thermopylae, I hope this picture reminds you of it. It's from movie 300 that's based on the story of the Spartans. And if you haven't watched it, I highly recommend it. And Lasonolidus because it's producing Lasonolide A. So I've been blabbering a lot for last 7 minutes or so. So let me summarize what I mentioned. Lasonolide A is a potential anti-cancer drug that's produced by Candidatus thermopylae lasonolidus. It's produced by a trans-city PKS pathway that we call LASPGC. LASPGC is horizontally transferred and is repeated 3 times, and the 3 repeats have some differences. And I just want to acknowledge and thank all these amazing people, especially the CONLAC, for all their help and support, and our collaborators, all from Florida Atlantic University. And I would love if you can contact me. I love collaborations. I love talking about bioinformatics, metagenomics. So feel free to shoot me an email or check out my GitHub. The story that you heard today is just a short snippet from our paper. It's on bioRxiv. Feel free to read it. And again, I'd love to chat if you have any comments. So thanks a lot.
Thanks a lot, Siddharth. It was a beautiful presentation. I want to open with a question. Have you thought about would you produce it large scale this Lasonolide? You're still in the let's say research stage.
I mean, so we can take the gene cluster that we have and put it in a plasmid, and then we can put the plasmid in a bacteria, let's say E. coli or strep. So that's basically got heterologous expression, and that could just increase its yield and produce it in a large scale. So yeah.
So it's already possible to do this. You're just still researching about which bacteria is the best for producing this.
So it's possible, but it's really hard. Like it's easy to, so people have put it in a plasmid. That's like my collaborators are trying to do it. But like to put it in the plasmid in a bacteria, that's a really hard process, the whole heterologous expression process. So they're trying it and we'll see. Fingers crossed.